It is visible?
Yes, sir.
Okay, wonderful. So we are going to talk about the research designing in Ayurveda, and we would be talking about what are the essential steps. So thank you again for giving me this opportunity to share my idea. So let us talk initially, what is the purpose of the designing? Why do we do the design? I mean, why at all a designing is needed? So we know that the designing is basically done to obtain certain specific function. I mean, whatever you design a machine, you design a motor car, you design your house, you design anything. What is the objective of the designing? The objective of the designing is to obtain some specific function which is related with that design. So everything, I mean, modern medicine says that it is the form which follows the function. So the primary objective of a design is what is that function? What is that function which you want to achieve from that design? And on the basis of that function, on the basis of that objective, the design has to be designed. I mean, whatever construct you are going to do, that has to be designed as per the specific function which you are looking at. So primarily the designing is to be done or is to enable the structure to achieve the desired function or the objectives. Moreover, this is also important that this enabling to achievement of the desired function or objective is to be done with minimum efforts and with minimum resources. That would be the perfect design. If you are able to do it, the same kind of objective, if you are able to achieve that objective with the minimum efforts and with minimum resources, possibly this would be the best design. We see that in Ayurveda, there had been lot of good designing done earlier. In surgery also, we see that number of instruments have been designed earlier. In Panchkarma also, we know that a lot of instruments have been designed. And we know that every such instrument in Panchakarma or every such instrument in surgery is basically aimed to perform some specific function. So again, I'm repeating that it is the function which was important, and the design was made as per the function. I mean, you were thinking of that, what is that function which you want to achieve, and as per that function, the design was made. And second thing, I mean, after seeing that why the design is important, we need to understand that how efficient the design is. Okay, you have designed it, now you have to see that how efficient your design is. So the basic thing which is going to understand about the efficiency of the design is that if it is requiring minimum resources, that minimum time is required, minimum money is required, minimum human resources required, how much ease of operation is there. If it can be done very easily, then all the efficient design. If it is workable in various conditions, then also it would be efficient design. If it is workable with various set of skilled people, it is not that a design should be workable with one very highly skilled person only. It should be workable with lot many people who are working in a similar area. So that would be the definition of the efficient design. And it should have minimum downtime, and it should have minimum chances of error, and it should also have easy correctability and repairability. So if these are things, these qualities are associated with the design, this would be that this is an efficient design. In research, how do you define in research, how we would be defining that what is the efficiency of the design? So for the simple definition of a research design, efficiency relates to the ability of a design to achieve the objectives of the study with minimal expenditure of resources such as time and money. So if you are able to achieve your objectives of your study with minimal expenditure of the resources, most important resources of the time and the money, then your research design is going to be said as an efficient design. Designing in healthcare, I mean, what we were talking about designing in general. Now we talk about designing in healthcare research. So what are going to be the first step to think of designing? I mean, the moment you think of that you are going to do some research, what are the questions which come in your mind before you start thinking of that, how you are going to perform that research? So the primary question which should come in your mind should be, what I wish to know. I mean, why at all you are doing this research? What is your research question? What is the primary thing you want to know from this research? This would be a sort of leading thing which is going to tell you or say you what research design would be most appropriate for that particular kind of research question. Second important question is how meaningful this question is in the context of the currently available knowledge and practices. Whenever you are finding a research question, you need to understand that what is the current context, what is the current knowledge which is already available. If you are going to repeat the same kind of thing which is already known by many people, possibly there is no sense of doing this kind of research. So, and moreover, this is also important to understand that how meaningful this question is. I mean, how meaningful your research is in the context of the current practices. If the people are already doing such kind of thing, then again, the kind of research which you are going to undertake would be questionable. Third important question is how meaningful this knowledge is for the common people. What it is going to give to the people. This research may be meaningful for you. This research may be meaningful for your institution or for your organization, but what it is going to deliver to the common people. So primarily the research should be aiming to deliver the benefits to the common people. So this is also one very important question which has to be there in the mind before you start or before you initiate any specific kind of research. And what are its translational possibilities? Whether your research is going to serve only the library or only the academic purposes, or it is having any translational possibility. So that is also something very important which has to be understood by every researcher. Now, a very tricky thing is this, that you need to understand that, do you have the resources to do that research? You might be having a very nice objective in your mind. You might be having a wonderful research question in your mind. But again, you have to think of that. Do you have the resources to conduct that kind of research which is there in your mind? Do you have enough of the time to do it? Do you have enough of the money to do it? Do you have the human resources to do it? Do you have the technical resources to do it? So if at all you have these all resources with you, possibly you can handle with that research. Possibly you can go with that research. And otherwise, if you don't have these all resources, there's no meaning of undertaking that kind of research because you don't have the means to go for that research. Another question is that, do I have essential? I mean, this is also very important, that do you have essential skills to initiate the research? So your own expertise is very, very meaningful. How much expertise you have to initiate that research, and what is the expertise of your team? So that is also very important. I mean, with whom help you are going to undertake that research. So unless you identify yourself in this background, you are able to answer these all questions, you won't be able to find a good research design for your study, because the research design has to give the answer to these all questions. What I wish to know, let us take these questions one by one and we'll be understanding that how these are going to be answered. So for primary thing, what I wish to know, I mean, why I am going to conduct this kind of research. So define grossly what you are aiming to achieve. The primary thing which you are looking from your research, it could be the primary objective. You have to understand what is the primary objective of your research. Also, you have to identify what could be the secondary objective of ancillary objective of the research. And you need to define very precisely what you wish to gain from your research. So for that reason, you have to draw your research question as elaborately as possible. Define your research as a research question, that this is the question, and by means of my research, I am going to try to find the answer of this research question and try to make it as elaborate as possible. So a design should precisely be made in order to achieve the objectives as closely as possible. So this is the first thing which has to be done by every researcher who is initiating any kind of research. Now, framing a research question is very important because the whole of the research is going to revolve around that. So you need to define very precisely something about the research, like what is that research, where this research is going to be done, why you are doing this research, on whom you are going to do this research, and how you are going to conduct this research. So these all questions are to be defined very precisely of the proposed research, and again be elaborate yet as precise as possible to define your research question. PICO is an acronym which is designed to frame a research question, which is consists of PICO, P stands for participants of a research or the population which is going to be taken up in the research. I stands for the intervention or the indications for the research, or if primarily it is for the clinical research, it is the intervention, what kind of intervention you are going to do. C stands for the comparator or the control, which you are going to undertake in this study, and O stands for the outcome. So if you have some idea about these four specific things, like what is P in your research, what is I in your research, what is C in your research, what is O in your research, you might be able to define your research question very precisely. So mind it that you have to find, or you have to design your research question on the basis of PICO analysis. Another very, very important thing is that you need to put your research in the context. In the context means where your research is standing in the background of the existing knowledge. So you need to understand what is known in the area, whatever area you have chosen for your research, what is already known in that area. For that reason, you have to do a thorough search of relevant literature. All the relevant literature you need to understand and need to look at. It would be consisting of the published research. It would be published or manually published research, unpublished research as well, or the classical text in Ayurvedic Samhitas. Experiences also, if you have an access to the experiences, then this all has to be searched so that you can get an idea that what is already known in the area. You need to understand what is unknown. What is unknown means the gross area where there are missing connections. You might be, after looking at that, this much knowledge is already available, and when you are sitting there in the clinic, you might be finding that, see, there is a gap. I mean, the patients are coming with this and this problem, and in the literature, I am not able to identify or find any specific solution for this particular condition. That simply shows that there are certain gross area where there are missing connections. So those missing connections are the areas which are unknown, and you might be willing to work for those areas. What are the relevant areas where knowledge is unavailable? This has to be identified. And again, you need to find that what do you, there might be a very, very wide area which is unknown, or there may be a number of missing connections. So you need to precise again, you need to be focused again, and you need to identify what you want to know. What do you need to know? And you need to put your research question in that context, and you need to see that how your research is going to help bridging this existing gap which you have identified. So this is the putting the research in the context. This is also very important. Another important thing is that what are the deliverables of the research, what you are actually going to deliver after doing or after conducting this research, what you are going to offer to the people. So you need to define very precisely that what are the primary deliverables. Primary deliverables means immediate deliverables, which you are going to give immediately after conduction. It could be the primary objective as well. There could be secondary deliverables or the remote deliverables. There could be secondary objectives, and there could be surrogate or surrogate deliverables also. You may call them the add-on deliverables or additional benefits which are going to be associated with the research. So the primary thing is to be identified, which is to be identified is what is expected to be delivered, in what time frame it is expected to be delivered, and what would be add-on benefits of the deliveries when you are conducting or when you are completing your research. So you need to identify that what you are expecting from your research. So this is also something important which has to be understood by every researcher. Now, when we are doing this research, we need to understand that for whom we are doing this research and what are the generalization possibilities of the outcome. You have conducted a research on certain specific set of the people, that is the sample. Now, what all observations you have made is related to that sample. You need to understand that how much applicable those all observations are, which you have observed there in a specific sample, to the common people, to the common population. So you need to understand that how much valid your research is for the population outside the research sample. This is very important because in your research sample, you have taken only a very, very small sample of the population. You need to understand that this observation which you have made for that specific population, whether it is applicable for the common people with a similar condition outside the sample, whether it is applicable for the common people with similar condition with comorbidities also, or to the marginal population with similar condition, to this population with similar condition. So this would be saying, this would be identifying that your research is valid not only for the sample which you have undertaken for the research, but would also be having a generalizability for the common people. So this would be making your research rather more translatable and more acceptable. Now you need to look at availability of the resources. That is the time, how much time you have. So we know that for the PG scholars, PG scholars, there is a limitation of the time. So you need to understand that how urgent it is needed to be done. Accordingly, you have to choose the research design. So how urgent is it is needed to be done? We see that in the current pandemic, COVID-19 pandemic, there are certain, it is quite urgent to finish with the research and to get the outcome of the research. So this is quite urgent. So you need to do something which is going to give you urgent results or immediate results. Emergency use of vaccines without proper clinical trials is one example of this activity. And in PG researchers and PhD, you know that it is a time-bound study program. You might be having 3 years or 5 years or 7 years to conduct your study. So you have to finish your study within that time frame. So your research design should be as per the time available to conduct that study. There can be a time-bound research project also. If you are doing a research for the research project, again it would be time-bound research. So you need to understand that how much time you are having in your hand, and accordingly the research design has to be made. Now another very, very important and tricky question: availability of the resources. What are the finances? How you are going to meet the finances which are going to be required for the research? Again, this is also going to play a very crucial role in defining the kind of research design which you are going to undertake for the study. Let us see what are what are those conditions where the finance is required for a research. It would be required for the cost of the intervention. It would be required for the cost of investigations, for cost of registration record, for cost of incentives to research participants, cost of extending awareness to invite the potential research participants, cost of compensation to research participant in case of any adversity if happens, cost of essential permissions if you want to have, cost of hired human resources if they are participating in the research, cost of hired technical services also you need to pay for, you need to have money, cost of data analysis also might be requiring some money, cost of publication also. When you are done with the research, if you are willing to publish your research, then journal will also be asking you to pay money. So for that also you might be requiring finances. So a lot of money is really required to do the research. So you need to, before you start doing your research, you need to understand that from where you are going to get the financial help. This is also one important thing. If you are willing to file IPR or patent, then also you need to put a lot of money for that. So the model of research finance, from where the research is being financed, it could be self-finance, it could be sponsored by pharma company, it might be done with the help of a research grant by funding agency, by contractual research also, you can get a contract to do the research. So this has to be identified. You need to, beforehand, you need to understand that from where the finances are going to be met. Now look at the resources. What are your resources? What is your research team? Who is going to help you in your research? So who else is there in your research team? You are the researcher, but besides that, who else is there in your research team? So research is to be done by a team, not by an individual. So you need to identify the right people for doing or helping that research. So you need to have a mentor or supervisor, the peers for the internal peer reviewing, a statistician you might be requiring, assessor in case of a triple blind trial. You need to have a data collector or the interviewer if it is a survey kind of a study. You need to have some technical assistant for the technical work if it is involved in your study. And you need to have, ultimately, finally, you have to have the research participants who are going to participate in your research. Another thing is you need to understand what is the availability of the resources, technical support, or how you are going to get the technical support. Technical support might be requiring at many places, like for the drug identification, for drug processing, for analytical chemistry, for heavy metal testing, for investigation, for data analysis, for subjective analysis, or you need to have some subject experts who are going to give you some idea about that research. So you need to identify beforehand what is your technical support. So that is also something important before planning a research. Individual competence is the most important thing, that what are your own credentials and qualifications, your own knowledge of the subject, your own knowledge of the basic research method, your own knowledge of the basic statistics. This is also very, very important. your aptitude of the research, your computer literacy. Many times we see that our students who are joining PG studies are not having the computer literacy, and for them it would be very difficult to perform a real research because they would simply be dependent upon other people for doing these kind of jobs. So you need to understand that what are your abilities, what are your competence, and accordingly you may be planning your study. You also have to understand that what is your team building capacity. Are you able to build a team for your work, and capability of work as a team? This is also very important, because again I'm saying that research is not being done by individual, but it is a kind of collective work which is to be done by a team, by a research team. Now we need to begin about the research design. Okay, we are going to choose the right research design. So the research design, you need to, again, we are going to reiterating what we have talked earlier. What is your objective of the research? Is it the safety or toxicity of a new compound on animals? Is it safety of a drug in healthy human volunteers, or it is dose-dependent safety studies, or therapeutic profiling of a compound, therapeutic efficacy of a compound, comparative efficacy of two compounds, observing a phenomenon in its natural setting? What you actually wish to observe? Simply, what is your objective of the study? So if you are defining your objective very clearly, possibly you would be choosing the most appropriate research design for your study. Common research designs and methods, you'll be looking after that. What are the common research designs and methods which are being adopted in the research, in the biomedical research, or in the healthcare research? This could be defined, or this could be classified as quantitative or qualitative research. Quantitative research, as the name implies, it is measuring the changes in the quantity. So because of that reason, it is being called as a quantitative research. And qualitative research, rather than or instead of measuring the changes in the quantity, they would simply be narratives, or they would simply be, I mean, if you are looking at your patient and if you are asking him whether he's feeling any difference from the treatment before he started the treatment and after he completed the treatment, it would simply be a narrative of the patient. He might be saying that yes, he's feeling better. So feeling better is also a research, but it is not a quantitative research, but it is a qualitative research, because it is a narrative which is being given by the patient. So that is also one kind of research which can be undertaken by Ayurveda as well. There could be interventional research, where the observer, or where the researcher is going to intervene inside, and intervene means they are going to modify the kind of the kind of behavior, or the kind of intervention, or the kind of anything which is related to the clinical setting, and to they are willing to observe the changes which are happening because of that intervention. Contrary to this, or in contrast to interventional, there could be observational research, where the researcher is merely observing that what is happening. I mean, he is not intervening anywhere, and he is simply observing the phenomenon which are happening around. So there could be this kind of classification also. There could be preclinical and clinical study. Preclinical study are primarily the safety and toxicity studies before the drug is being used for the humans, or the human study. And the clinical study are primarily the human studies which are being done upon the human beings or human volunteers. There could be randomized or non-randomized studies. This depends upon if you are adopting the randomization methods or not adopting the randomization methods. And these are the methods primarily which are being utilized for the reduction of the bias. Similarly, there could be the blinded study or the open study. Blinded study means you have blinded the people and they are not known about that what kind of intervention is being provided to them. There could be, we are going to talk further, there could be single blinded or double blinded or triple blinded, depending upon how many people are blinded for a study. And contrast,
आणि ते कॉल केले होते खालून.
It could be open study.
अगं तुम्ही कापले दीक्षा, मला कंटाळा आला होता मोठ्या केसांचा. आणि माझी
Dr. Nitin, there is somebody speaking. Can you please ask them to stop?
फ्लिक्स कट करते ना इथून, ते मस्त दिसते. श्वेताने फ्लिक्स केलं.
All please keep their mic on mute.
समोर एवढा भाग एवढा येते, आणि मग खालची स्टेप विथ लेअर केली आणि हे ते फ्लिक्स टाकलं.
Dr. Nitin, please.
Yes, I am telling Dr. Vijay sir, please can unmute, can mute all of the speakers?
Muted.
Okay, sir. So we have muted all the speakers, all
Okay,
the other
okay,
participants.
Thank you. Now, looking at the quantitative and qualitative research designs, there are some common quantitative research designs. Quantitative, as we already have talked about, quantitative are those kind of studies where the changes are measured in the quantities. So this could be the experiment or the cohort studies or the case control studies or the randomized controlled trials. These are the examples of the quantitative research designs. Contrasting to this, there could be the qualitative research design where the changes are not measured in the quantities, and these are simple narratives which are being identified by such researchers. So these could be the surveys or the interviews, observation of the participants, or sometime it happens, it may be done with the secondary data analysis also. Observational studies, there could be two kind of studies. One is cohort study and another is case control study. Cohort stands for a group of the people. So cohort study is usually a sort of prospective study where we have identified two kind of cohorts. One cohort is that kind of cohort which is exposed to certain kind of risk factor. For example, let us take an example that if we are willing to identify that a cohort which is exposed to the cigarette smoking is prone for the development of the lung cancer. So this could be a wonderful cohort study. If you want to do this kind of a study, you have to identify two population. One population is that population which is exposed to the risk factor. That means who are the smokers, who are the cigarette smokers. At the same time, you have to identify another population which is not a smoker. And you have to follow up these two populations in the prospective future. In the future time, you have to identify how many among the exposed population, I mean, those people who have been exposed to the risk factor develop the disease or the final outcome, whatever you have decided to be the final outcome, develop the disease, and how much of them have not developed this outcome despite of being exposed to the risk factor. And similarly, the same kind of exercise you need to do with the people who are not exposed with the risk factor. There is a possibility that some of them might also be developing with the final outcome or the final disease which you are looking at. And again, similarly, there may be some people who may not be developing this kind of outcome. So this is going to be the cohort study. This is very important because you might be seeing that cohort studies usually, because they go in the future, they are prospective studies, and usually they take very, very long time. I mean, you might be exposed to the risk factor now, and you might be developing a disease maybe 10 years later or 20 years later. So here comes that how much important the time is. I mean, if you are having a 3-year time period in your hand to conduct your study, possibly you cannot do a cohort study. So this is the importance of research designing, considering all those factors which we have talked earlier. This is another example of the observational study, case control study. Case control study, in contrast to the cohort study, this is usually a retrospective study. We have identified the cases. Cases are those people who are already developed the condition of interest. I mean, they might have developed a disease. They might have developed a condition which is of our interest. And the controls are those people who not have developed such conditions. So now we are going to look back in the retrospective phenomenon. We have to identify that how many of the cases have been exposed to the risk factor earlier, and how many of the cases have not been exposed to the for the risk factors in the history, in the retrospective means. Similarly, we have to identify the same kind of phenomenon with the controls also. So by this way, we would be identifying that what is the relative phenomenon of developing this kind of specific disease condition among the people who are exposed, and what are the possibilities of developing this kind of disease condition among the people who are not exposed. So this is also a wonderful research study. This might be requiring less of the time. So this can be done. Case control study can be done within 1 year or within 2 years. So if you are having limited time period, possibly this would be a wonderful study to conduct upon. Now look at the qualitative research. Qualitative research, there can be a number of researchers, for example, the in-depth interviews, the focus group, the projective technique, the case studies, ethnography, observations. A lot of qualitative researches are being adopted in the modern medicine. There are two researchers which are of importance. One is action research and another is grounded theory. Action research is a series of, basically these two researches are being adopted in the sociology, and they are not really very much adopted in the biomedicine, but they have some application in Ayurveda. You can do something in Ayurveda. So what the action research is basically, I mean, you have started doing the action and then you are following it by the research. You might be understanding that what this action might have given to the people. So the best example of this action research is projection of the healthcare workers during protection of the healthcare workers during COVID-19 by the Ayush Path. I mean, the moment when Ayush Path was being distributed among these people, there was no idea that how this is going to be done and how this is going to protect the people. So the primary idea was the action. Primary thing which was done initially was the action. You have started doing it. And then gradually, when you found that, yes, it is helping, you have started identifying that how it might be helping the people. So this is a wonderful example of the action research, which has its applicability in Ayurveda. And the grounded theory research is another example of the qualitative research. This is the research which is aiming to generate the theory. So it begins with a broad query in your mind. I mean, you have certain big question in your mind, and you start collecting the relevant information from what all sources these information are available. And what you do, you review all the information, you compare the information available, you contrast the information, and then you summarize. And then finally, you find it out that what all relevant information is ultimately saying about. And by means of this constant comparison process, you might be identifying the commonalities and the dissimilarities among the categories of information, and you might be reaching at that how this thing, whatever you are trying to find, is functioning or is working. Now comes the interventional and observational research designs. Interventional study, as we have talked earlier, interventional study is that where the researcher is trying to intervene inside the research or intervene in the process of the treatment. It could be experimental, and it could be further defined as further divided as randomized controlled trial or non-randomized controlled trial, or lot many different kind of studies can be done under the experimental or the interventional study. And if you are not intervening, if you are not doing anything and you are simply observing what is happening, it would be the observational study. There could be the analytical observational study. There could be analytical observational study as is being done in the case control study and the cohort studies. And this could be descriptive study also, as might be done in the in-depth interview or the focus group discussions like kind of thing. Preclinical research designs also are there, depending upon what your idea is of the research, why you are going to do. So goal of the preclinical study is to identify initial safe doses and dose escalation schemes in human. This might be identifying target organs for the toxicity, might be studying the toxicity and to see whether this toxicity is reversible or not. And it might be identifying safety parameters for the clinical monitoring. So this is the primary study which is going to be done if at all you are trying to make some research on a newer molecule where a lot of information is not available, or information related to these things is not available. There can be a number of ways to do the preclinical researches. It could be in vitro. In vitro means it could be done with the microorganism, with the bacteria, or with any microorganism. It could be done with the cell also, or it could be done with the biological molecules. These days, a lot of studies related to the COVID-19 have been done on the biological molecules. We call them molecular docking studies or in silico studies. So a lot of such studies can also be done. And these are the preclinical studies because they have not yet been tried upon the human beings. So for that reason, it is being called preclinical research. This could also be done in vivo. In vivo means it is done inside the living organism or in the vicinity of the living organism. And most commonly, the living organism which are being chosen for such studies are the animal models. So in vitro is used to describe work that's performed outside of a living organism. In vitro, no, I'm sorry. In vivo is done in the human being, in the animal model. Okay, now we talk about the clinical study design. Clinical study design could be the trials, could be the experiment, and could be the observations involving human beings. This is a very, very fundamental thing. The moment you talk about the clinical study, this essentially involves human beings. And it might be a trial, it might be observational, it might be experimental study, it might be interventional study, in whatever kind you do. But the basic and essential thing is that you are involving the human beings for such a studies. So again, there can be a number of ways, number of designs to do the clinical trial. Clinical trial could be observational, could be interventional or experimental. Observational can be divided in multiple sub-classifications, subdivisions. It could be descriptive. For example, the case report or the cross-sectional studies or the case series. These are the describing individual cases or a series of the cases. And it could also be analytical, as we talked about. This could be the case control studies or the cohort studies. And it could also be the meta-analysis. Now, we need to look at how we are going to reduce the bias and the error in our research. So there are a number of ways and number of means which are going to help you reducing the bias and error in your research. So this could begin with the sample. You have to have enough sample which is going to be representative of the real population. So sample technique is very, very important. If it is a very selective sample, possibly you might not be getting the proper inferences from your observation because your sample is not the representative sample. So that is very important. And the second important thing is the sample size. If sample size is very, very small, possibly the outcome which you are going to observe through the research might not be very reliable. So to have the study effect, you have to have a bigger sample size. So that also defines the quality of the research. Another method to reduce the error or to reduce the bias is the randomization, which could be true and false randomization. We need to understand many times we have seen in Ayurvedic researchers that the people arbitrarily write that they have done randomization without having a real idea that what randomization is. And when the moment you ask them that how you have done this randomization, they might be simply saying that they have done a coin toss or they have done something like that, or they have done a kind of sequential randomization. They say that, okay, the first patient which came, they have given the treatment A, the second patient when came, they have given the treatment B, and this is how they have done the randomization. Unfortunately, this is the pseudo randomization, the false randomization. So you need to understand what the randomization is and how it has to be done in a clinical research. Third important thing is the blinding. Blinding is done to mask the effect of the medicine itself or the physician itself. So it could be the single blinded, it could be the double blinded, it could be the triple blinded, depending upon who is actually blinded. And finally, you can have the testing for the reliability and validity. You might be utilizing a lot of parameters in your research. In Ayurveda, it is very commonly being done. We utilize a lot of subjective parameters. For example, we have a lot of clinical information related with the disease. We have identified a set of signs and symptoms in relation to that disease, and we arbitrarily have developed a sort of parameter to determine that how this treatment is giving the responses in a particular patient in reference to that particular parameter. So before you utilize these parameters which you have designed for your own research, you have to see whether these parameters are really reliable and valid or not. So unless you make them reliable, unless you make them valid, there is no meaning of utilizing those all outcome parameters, because your research on the basis of those outcome parameters is going to be challenged for sure. And the people are going to ask that whether your parameters which you have utilized for your research have been validated or not, or whether they have tested for their reliability or not. So you need to understand that how the reliability and validity has to be done for your research. Again, we are going to talk about the blinding, blinding contrasting to the open design. This could be the single blind, where we see that the patient is blinded and the physician is knowing that what kind of treatment or what kind of intervention he is going to give to the patient. So there could be, this is going to reduce the bias from the perspective of the patient. If the patient are aware that they are going to receive this and this treatment, there might be a possibility of change in perception, and they might be having a sort of influence by the kind of intervention. And this happens very commonly in Ayurvedic clinical researches. For example, I'll give you just one example. Many times we do Sanshaman Chikitsa along with this Sanshodhan Chikitsa. And we do, we know that take one example of the arthritis patient, osteoarthritis knee, or the Sandhivat patient. If you are recommending a patient to go for the Nadi Swed, and if you are recommending a patient to go for a Sanshaman Chikitsa only for the medicine, there is a strong possibility that the people who are receiving the Nadi Swed might be responding more nicely comparing to the Sanshaman Chikitsa, because it may not be, there may not be a real difference, but the difference is of the procedure, or difference is that they are knowing about the procedure. They are seeing that something is being done upon their joint, and this is going to help them feeling better. So there might not be a real difference between these two things. So this is really going to give difference of the observation when you finally conduct your study. There could be the double blind, where the physician is also blinded. Physician also do not know that what kind of treatment is going to be offered to a set of the patient. And there could be triple blind also, where the assessor, assessor could be the third person who is going to analyze the studies. So he is also being blind. He also don't know that group A belongs to which kind of intervention and group B belong to which kind of intervention. So this could be the triple blind. So the double blind studies are most precious studies which are being considered there in the modern medicine. But unfortunately in Ayurveda, the blinding is one of the most trickiest kind of things, because a lot of things which are being utilized in the treatment in Ayurveda are quite obviously known. They are quite obviously known. They are quite obviously understood by the patient that they are receiving this kind of treatment. So blinding is really difficult kind of thing to be adopted in Ayurveda. Now there comes the controlled and uncontrolled design. So what is controlled and uncontrolled design? We have talked earlier in the PICO analysis that C stands for the control. So whenever you do your study, whenever you provide some kind of intervention to your patient, you need to understand that this treatment, whatever treatment, there might be some kind of efficacy of your treatment which you are offering to the patient, but we need to understand that how much this treatment is effective in comparison to the kind of other treatment which are already available for that particular kind of condition. So for that reason, we have to understand that we need to find some kind of control with whom you can compare the observation which you have done with your intervention. So there can be number of controls or number of comparators. It could be placebo, where you are not giving the patient or the group any pharmacologically active compound. You know that this is not having any pharmacologically active substance inside it, and it is not having any pharmacologically active property. So that would be the placebo. So you can control, or you can compare with the placebo. You can compare with the no treatment. You can compare with the dose response. You might be giving the dose, and then you can increase the dose and you can compare the response. So it could be the dose response control. It could be the active or positive control. Active or positive control means you are providing them another set of intervention which is already being given to the people of the similar condition. This might be the standard therapy. Many times we call about that. It could be the standard therapy which is being offered to such kind of patients, or it could be any kind of active treatment which is being offered to those kind of, or those set of the people. And it could be external or historical control. External control is the kind of control which is outside your group. You can compare your treatment outcomes with a kind of group which is having no control with your study. It is not involved in your study initially. You are having no control with that, but you are simply comparing it. For example, you are treating a group of the diabetes. You are giving the treatment to them, and you can compare the observations which you have done on your study, which you have observed in your study, with a kind of external group where you are not offering any kind of thing. You are not having any control upon them. They are receiving whatever they wish. They are choosing any specific kind of diet they want to wish, and they are doing at their own. So you do not have any control upon them. So this would be the external control, which is beyond the control of the researcher. So this would be the external control. And there could also be historical control. Historical control is also very valid for Ayurveda. If we are having a good good habit of the data keeping practices. This could be the hospital data. Hospital data means you already have some data there in the hospital where the similar kind of patients have already been registered or have already been treated earlier. So their data is preserved in the hospital. So whenever you are doing some new research, you can compare your observation with the data which is already available in the hospital. You can see that what was the outcome of those who have earlier been treated with some different kind of intervention and with the kind of observation which you are providing them today. So this could be the external, this could be the historical control. So unless you have a control or unless you have a comparator, possibly your research might not be that much valid if you have a control to compare your observations. So most of the times when you look at the Ayurvedic researchers, you would be finding that almost, I mean, we won't be finding any control in Ayurvedic researchers. So what we do, we usually do, there is no control. What we call them pragmatic trials or the exploratory trials. And we simply do it in a single group. We do a pre and post intervention comparison, and it is being done on the same set of the patients. So unless you have a kind of comparator or unless you have a kind of control to compare with, unfortunately, the observations which are being made in a pre and post intervention, they are not really very much valid. There are specific conditions where these controls are to be used. External control, we already have talked about. Historical control also, we already have talked about. Other study designs which are relevant to Ayurveda, there can be. I mean, these all with what study design we talked about, these are the study designs which are being considered in the modern medicine. And these are very much used, but they are very much useful for the Ayurvedic research as well. If your resources are permitting you to go for those kind of researches, you can definitely choose some of those kind of study designs which are already proven to give those kind of responses or those kind of observations. Besides this, there are some certain specific designs which are relevant to Ayurveda alone. There could be the single case design. The patient is his own control. This simply says that you are treating a patient longitudinally and you are observing the patient for quite long term. For example, I mean, take an example of a diabetes patient. You might be treating the diabetes patient with certain kind of intervention. And after a certain period of time, you have treated him for 3 months or for 6 months, and then you have observed that diabetes has not been controlled. You have changed the treatment and you have provided another set of the treatment. And again, you are watching the same kind of patient, same patient for another 6 months. And again, you are identifying that what your observations are. And similarly, you can change, you can multiply, you can manipulate the kind of treatment, and you can observe the same patient. I mean, it is a single patient. We call it N of 1 trial. So this patient is observed longitudinally in long run. You can observe the same patient for years together. I mean, for many years you can consider, and you can be watchful upon that patient, that what is happening on that patient. And ultimately, if some kind of benefits are being observed, you might be seeing that what actually has been given to the patient, which has given the benefits ultimately. There could also be the black box design, which is also very commonly utilized in the Ayurveda. Black box design is a kind of whole system research. Many times it is being told in Ayurveda that, see, in Ayurvedic research, when we try to do a research on certain specific component of the intervention, many times, most of the times, we are the clinicians and we do the practice also. When you provide some kind of intervention upon your patient, you know that a lot of things are being provided to the patient. There could be the dietary recommendation, there could be some kind of lifestyle modification recommendation, there could be the treatment, there could be Sanchaman and Sanshodhan therapy. Both of them might be utilized in the same kind of patient. And there could be number of therapies also. I mean, there could be number of formulations which are being utilized. So this is the real practice. This is the real practice of Ayurveda. So when actually we are utilizing this kind of practice, this would be very hard to identify if one specific component of that research or that intervention which is being done, which is being provided to the patient, is going to give any kind of benefit to the patient. So for that reason, it is being considered that, see, Ayurveda doesn't work in components. Ayurveda works in a holistic manner. So unless you combine all of them together, unless you combine it pathya and apatya, unless you combine the dietary recommendations, unless you combine it with the lifestyle recommendation, unless you combine with multitude of the interventions which are being provided to the patient, you won't be able to get the proper responses. So for that reason, it is being considered that, okay, fine, you do whatever you do in your clinical practice, and we would be considering it as a package. We would be considering at a package, and we would be thinking of how was the state of the patient before the package was given to the patient, and how was the state of the patient after the package was received by the patient. So instead of considering and instead of evaluating a component of the research or component of the intervention, we would simply be evaluating the whole package. So this is some specific research which is being adopted by Ayurveda. WHO call it as black box design. There can also be the ethnographic designs. This is the hypothesis generation on the basis of the folklore practice. Many times it happens that when you talk to the people who are practicing in the remote areas or in the tribal areas, they might be having certain idea of their own, and they might be thinking of that this drug or this particular kind of compound is working in this such and such area. So to explore this thing, you cannot go for some specific kind of research. You have to generate a kind of hypothesis. You need to sit there with those people. You need to understand that what they are doing and how they are defining it, how they are identifying the drugs, how they are identifying the disease condition, and how they are identifying that in this disease condition, this particular drug has to be used. So if you are looking at, if you are willing to identify such things which are prevalent there in the folklore practices, you need to identify, you need to adopt the ethnographic design. And observational designs are the wonderful designs which are to be adopted by the Ayurvedic people a lot, because I mean, they may not be having any intervention, but with the other aspect of the disease. But if these are being done honestly, and these are being done meticulously, possibly they are going to be as good as the clinical researchers or as good as the randomized controlled trials. So let us summarize. I'm going to finish. There are certain points which we need to understand. Research design is essential to be thought of before initiating a research. We need to understand that after initiating the research, there is no meaning of talking about the research design. So the moment you think of initiating a research, you need to think of that. What is that research design which is going to fulfill your objectives which you have in your mind? Research design follows closely the objective of your research. Research design is modifiable as per researcher's individual capacity. If you have certain kind of limitations, possibly you need to modify the research design accordingly. The result of a research are weighted against the research design used in the study. This is very, very important. I mean, you might be putting an observation, but the observation is going to be weighed against the research design which is used. So this is the reason that the modern medicine also we say that randomized controlled trial, if it is being done, if your observations are done through the randomized controlled trial, the results are valid. And if any other kind of research is being done without the rigors of the science, possibly whatever observations you have made, you have said that this treatment is very much effective or this treatment is not effective, nobody is going to bother about your research or results, because they might be thinking of that the design was not perfect to have these kind of observations which you have made in your research. And choosing an appropriate research design is as important in Ayurveda research also as it is there in the modern medicine. So whatever research you do Ayurveda research, you do modern medicine research, there are certain parameters to do that research, and you have to choose an appropriate research design if you are going to do it perfectly. Thank you very much. Dr. Nitin. Dr. Nitin.
Sanjeev, please unmute.
Dr. Nitin.
Manish, I think his voice is not. Okay. Thank you, sir, for your excellent talk. I can say I can very elaborately you given all the points. Although it is a very tough and very vast topic, as you said earlier. And now we are moving towards the question hour session. which has to be conducted by Dr. Jaya. Are you listening? Please unmute and
Okay, sir. Thank you,
go
sir.
by the questions.
Thank you, sir, for inviting me. First of all, I extend my gratitude to everyone and convey my regards to all teachers and seniors present over here. And special thanks to Dr. Rastogi, sir, such an eminent personality. And I'm sure that all of us have gained much from today's session. But our listeners are so curious that they still have many more doubts to ask. So I request you to, sir, kindly take those queries and clarifies those doubts. First of all, first query is from Dr. Neeraj Jain. He asked that what is effect of Agni Karma on sciatica? One second, sir. From Dr. Neeraj Jain. He wants to know that study the effect of Agni Karma on sciatica. So how the study should be planned? Will it be classified in qualitative research, as only patient will tell that I have relief, or it will be quantitative, as doctor will analyze that by doing SLR test and all. So kindly clarify that, sir.
See, this is my humble request to all the participants that you might not really be able to understand a lot of terms which are being utilized in the research methods and designing just in one session. This is really going to take much of the time to understand what are the qualitative researchers, what are the quantitative researchers. Just to distinguish between these two, as I already have told, that quantitative researchers are those researchers where the changes are going to be measured. If there is a measurable quantity, in sciatica also you can do it. If there is some certain kind of pain which you are able to measure, you can measure it through the VAS scale or visual analog scale or any other kind of measurement. If you are able to adopt that kind of measurement, and that if you are going to put in your research as a kind of outcome parameter, it would definitely be a quantitative research, because there is certain kind of quantity which you are going to measure for your research. But if you are simply observing that, yes, patient got benefited, I have given this Agni Karma to the patient, and the patient is saying that he is fine, his pain is gone, and this and that. So this would not be a quantitative research, because you didn't quantify anything. And you are simply taking the narrative of the patient, and the patient is saying that he got benefit from the patient, from the therapy. So this would be a qualitative research. Got it?
Yes. Yeah, sir. Thank you, sir. Nicely explained, sir. Second question from Dr. Karisma Varlani. She asks about what is in silico or molecular study.
This is something tricky. I mean, Ayurvedic people have nothing to do, because this is a domain area of the molecular biologists. So you need to not understand that what is the docking study or in silico study. Primarily, this is a kind of a study which is being done on the biological molecules, and to see that how these molecules might be acting in a particular kind of condition, how they might be acting on the viruses. A lot of studies are being done. Molecular studies have been done currently on the COVID-19. I mean, this SARS-CoV-2 virus. A lot of in silico studies have been done. But these are the specific studies which are being taken up by the molecular biologists, and Ayurvedic people have nothing to do with that.
Okay, sir. Thank you, sir. Dr. Neeraj Jain again asked that another doubt is that when we work on hospital, all the patients will not come together. So how to design study?
I didn't get the question. I'm sorry.
It
is quite natural that all the patients will not be coming in the hospital at one time.
Yes, yes,
so,
sir.
I mean, what it has in relation to the research design?
Yeah, irrelevant question.
Research design is a different thing.
Okay, sir. Thank you.
There might be the patient. There might be please.
Yes. Next question from Archana Baranwal. She asked that how to randomize study. Already you had explained earlier, sir. Yeah. But again, she asked that how to randomize it.
How to randomize this study? The simplest method is there are random tables, there are computer generated random tables. See, what is the meaning of the randomization and why should we do the randomization? I mean, if you have this idea, you might be knowing that how to do it. So randomization is primarily done to create a kind of homogeneity between two groups. For example, if you are having a comparative study, you are having two groups in your hand. There is one group where you are going to give one kind of intervention, and there is another group where you are going to give another kind of intervention, and you want to make the comparison of that. So the primary thing is this, that unless there is a kind of homogeneity between these two groups, just take it very seriously what I'm trying to explain.
Yes, sir.
There is the basic difference between these two groups. For example, the group A belongs to more serious cases comparing to the group B, which are having the mild to moderate illnesses. Whatever kind of intervention or whatever kind of control you are going to provide, there is a certainty that the differences would be there, not because of the intervention, not because of the control, but because of the primary condition, primary health condition which the patients were having. Because you already have selected a group which is having more severe kind of conditions comparing to the other group which is severe condition. So these two groups are not homogeneous. So they are not comparable also. When these two things are not similar, how can you compare them? So randomization is very, very essential to make them comparable. This is the primary thing. Unless you randomize them, you cannot compare them. So this is something very important. So randomization simply says that whatever you do, whatever kind of intervention or whatever kind of thing you are going to offer, the physician or the patient should not be known of what kind of treatment he is going to be recommended. Whether he is going to receive the placebo, whether he is going to receive the intervention, whether he is going to receive the standard care or whatever, what all kind of comparator we have talked about, they should not be known about. If they are knowing about, this would not be the randomization.
Okay, sir. Nicely explained, sir. Next question is from Dr. Nisa Gupta. She again asks, what is action research and grounded theory? Sir, kindly explain again in short.
Dr. Sanjeev, we already have talked about this. Action research is simply like action is the first thing which is being done. Mostly what we do in the research, we make the research, we do the research, we do the hypothesis, we identify certain observations, we make the results, and then we follow them, we take them into the action. I mean, first you conduct the research and then you do the action. In the action research, action is primarily done. I mean, you first started doing the action. You have started doing something and then you are analyzing, then you are doing the research that how this action might have helped in that certain kind of condition. So this is the action research. And this is commonly being done in the sociological research. This is very less adopted in the biomedical research or in Ayurvedic research. And similarly with the grounded theory.
I think Dr. Nisa has understood properly. And next question is from Dr. Vaisali. Her question is that how many sample size should be taken to start research?
How many?
How many sample size should be taken to start the research?
Sample size calculation is also a very, very tricky thing, see, and it depends upon 2-3 fundamental things. One very fundamental thing which you have to understand is that how common that disease is. If the disease is very common, naturally the sample has to be very big. If you're doing a research in diabetes, possibly which is having 7 to 8% incidence ratio or prevalence ratio in the country, possibly you need to have a very, very big population for the research or very big sample for the research comparing to a condition, a clinical condition, which is very rare, which is not very easily available. If you are asking about certain kind of clinical condition where a very rare kind of people are available with that kind of research, you won't be able to get that kind of sample. So this is one thing. You need to understand that what is that incidence? What is the incidence of that that particular disease upon whom you are going to research? This is one thing. Second thing.
Okay, sorry to hinder you, sir. Continue.
Okay.
Sir.
The second important thing is this, that you need to understand what is the effect size. What is the effect size means? You have to look at the previous researchers and you have to understand that what was that quantum of benefit which has already been done by the earlier researchers. For example, some study has already conducted earlier and they are showing that 20% improvement is there or 30% improvement there is there. You need to consider that also in sample size calculation. And thirdly, third important thing is that you need to consider or you need to understand what are the possibilities of the error in your design. So how much error you are going to do? If more error is there, bigger sample is to be needed. If a small error, if less error is there, you can go for the small sample.
Okay, sir. Okay, sir. Thank you, sir.
Okay.
Next question is from Renu Rathi. She asked that about what is true randomization. Please give your insights, as what you have explained, false is a type of randomization. What is true randomization? And you have explained earlier that what is false type of randomization. So give your insight about that.
True randomization, if you talk about true randomization, is that kind of randomization where every research participant, whosoever is participating in your research, should have equal opportunity to be given any kind of intervention, any kind of, I mean, they can be allocated with the placebo, they can be allocated with the intervention. So they have the equal opportunity. So there is no bias in the physician's mind, or there is no bias in the researcher's mind. So we are not prioritizing the patients on the basis of anything. We are not prioritizing the patient on the basis of the disease, on the basis of their clinical condition, on the basis of their willingness. Many times it happens that you recommend a patient for the Vaman or for Virechan, and many times patient refuse to go for the Vaman. And they say that, nahi, doctor sahab, koi khali dawai likh dijiye, koi hum nahi jayenge uske liye. Theek? So what you do, if you have two groups in your mind, what you simply do, you simply say that those who are not willing to go for Vaman, you put them in another group. And those who are willing to go for the woman, you put them in another group. So this is wrong. This is the false randomization. This is the false randomization because you have not given them equal opportunity. They should be randomly allocated. Randomly allocated means you do not know that to which group this patient is going to be. I mean, which group he is going to be allocated. He could be allocated with the intervention. He could also be allocated with the placebo, or he could be allocated with the standard care, or whatsoever comparator you are going to use. So this is also, this is very important, and this is the true randomization. And if at all you know, for example, if you make a toss, or if you put it in the sequential manner, you put A and B, A and B, A and B, you give the first patient A and the second patient B, so then everybody knows that the next patient who is going to come to you is going to be allocated with that kind of treatment. So I know, and the patient would also be knowing, because the earlier patient has received this treatment. So I'm going to be there and I'm going to receive this kind of treatment. So this is false randomization, right?
Okay, sir. Okay, sir. Understood, sir. I think two more questions are there. One question is from Dr. Mrityunjay Dwivedi. He asked that what will be the best sampling technique and what should be the best sample size to perform a study on a list of books mentioned in CCIM curriculum, and please suggest some ways to maintain the randomization in such studies.
I need to take a full lecture for that to answer the question.
Okay,
Just to summarize, see, sample is what? Sample should be the representative of the population. Whatever sample you are going to choose, it should not be a selective sample. If you are choosing the people from certain specific group, for certain specific area, for certain specific population, this would not be representative. So sampling should also be a random sampling. The best technique of the sampling should be the random sampling, right? And the sample size calculation we already have talked about. Sample size calculation, I mean, considers a lot of things. So you need to put all these things for the consideration of the sample size calculation.
Okay, sir. And last question is from Dr. Iswar Dutt Sharma. He asked that in which category the Rog Nidan principal research categorized like Nadi Pariksha with respect to any diseases?
I didn't get the question.
Should I,
Sorry.
okay, sir, should I, in which category the Rog Nidan principal research categorized like Nadi Pariksha with respect to any diseases?
What is the question? I'm really
I
sorry.
think the
I
question
am
is is
Yeah,
not
sir,
written
means,
correctly.
yeah, sir. Means I think he asked that Nadi Pariksha research category like Nadi Pariksha. Okay, sir, let it be. So I think all questions I have taken, and 2 to 3
Madam,
questions.
if time permits, can I ask one question?
Yeah,
please,
yeah,
please.
sure, no problem.
गुड इवनिंग सर. सर मैं ये पूछना चाहता हूँ कि स्टिल जो स्टैंडर्ड रिसर्च है, क्लिनिकल एक्सपेरिमेंटल रिसर्च, वो अभी भी डबल ब्लाइंड प्लेसिबो कंट्रोल्ड स्टडी को एक्सेप्टेंस है. तो हम उस प्लेसिबो को इंटरनल मेडिसिन इन आयुर्वेदा, अगर हम किसी इंटरनल मेडिसिन अपनी दे रहे हैं तो एक प्लेसिबो को किस तरह से हम यूज कर सकते हैं, क्योंकि कोई ना कोई प्लेसिबो हमारे यहाँ या तो जैसे हम हनी देते हैं या फिर कोई सिरप देते हैं, और एग्जैक्टली प्लेसिबो शुड बी एग्जैक्टली द in the same formulation, in the exactly it should be the same looking type, so that randomization should be true, there should be true randomization. So क्या सर हमें अपने आयुर्वेद रिसर्च में प्लेसिबो को स्टैंडर्डाइज करने की जरूरत है, या फिर डबल ब्लाइंड प्लेसिबो कंट्रोल्ड स्टडी ही एक गोल्ड स्टैंडर्ड होगा, या जो हम इस तरह की रिसर्चेस अभी कर रहे हैं कि सिंगल एंड स्टडी, वो भी स्टैंडर्ड रिसर्च मानी जा रही है आजकल, या वो भी ठीक है? सर मैं इस तरह में अगर आप समझ पाए तो.
डॉक्टर साहब देखिए, स्टैंडर्ड रिसर्च तो वही है जो डबल ब्लाइंड रैंडमाइज कंट्रोल स्टडी ही है, आपने वही स्टैंडर्ड है, और आज भी उसको वो गोल्ड स्टैंडर्ड ही बोलते हैं. हालांकि उसकी अपनी लिमिटेशंस हैं, उसकी अपनी प्रॉब्लम्स हैं बहुत सारी. आयुर्वेद के साथ ये प्रॉब्लम है कि आपने बहुत अच्छी बात बोली कि प्लेसिबो बहुत आसानी से हम डिजाइन नहीं कर सकते आयुर्वेद में. हम शायद इसी वजह से ये काम नहीं कर पाते हैं, क्योंकि प्लेसिबो डिजाइन करना, प्लेसिबो में जैसे आपने कहा कि इट शुड लुक अलाइक, इट शुड बी कंप्लीटली सिमिलर टू द इंटरवेंशन. आप जो भी इंटरवेंशन दे रहे हैं वो पूरा का पूरा उसके जैसा ही होना चाहिए, तभी वो प्लेसिबो होगा. तो क्योंकि रोगी को ये आभास होगा कि हाँ वो उसको दवा मिल रही है, रोगी को ये नहीं पता लगना चाहिए कि उसको प्लेसिबो मिल रहा है. तो ऐसा करने के लिए हमें पूरा एक जैसा ही उसको बनाना पड़ेगा. अब अनफॉर्चुनेटली आयुर्वेद में क्या है, आप बहुत सारे प्रोसीजर यूज करते हैं जिनका प्लेसिबो डिजाइन कर पाना संभव नहीं है. प्लेसिबो डिजाइन कर सकते हैं कहीं कहीं, किसी जगह कर सकते हैं. जैसे एक छोटा सा एग्जांपल दूं, प्लेसिबो कैसे बना सकते हैं. आप प्लेसिबो जलौका अवचरण करते हैं, जलौका लगाते हैं, जलौका दिखाई पड़ती है सामने आदमी को, आप सामने के हिस्से में कहीं पे लगाएंगे, जलौका लगाएंगे, उसको पता लग जाएगा कि जलौका लगी हुई है. सपोज करिए हम किसी ऐसी जगह पे जलौका लगाते हैं जहां पे उसको दिखाई नहीं पड़ती, पीठ में आप जलौका उसको लगाते हैं. आपने क्या किया, आपने जलौका का पात्र रखा उसके सामने, जलौका भी रखी वहां पे, सामने दिखाई पड़ रहा है कि जलौका कर रहे हैं आप, लेकिन आपने जलौका नहीं लगाया उसको. आपने किसी और तरीके से उसको महसूस किया, उसको दिखाई नहीं पड़ रहा है, उसको ये लग रहा है कि हाँ उसका ट्रीटमेंट हो रहा है. ठीक है, ये क्या हुआ, ये प्लेसिबो हो गया, ये सीधा प्लेसिबो. तो ऐसे करके आपको बहुत सारी जगहों पे जहां पे आप आयुर्वेद में रिसर्च कर रहे हैं, आपको ये ढूंढना पड़ेगा कि आप इसमें किस तरीके का प्लेसिबो इस्तेमाल कर सकते हैं, कैसे बना सकते हैं प्लेसिबो, है ना. जहां पे नहीं प्लेसिबो बना सकते वहां पे आप दूसरी तरीके से रिसर्च करिए. प्लेसिबो कंट्रोल स्टडी में एक और बहुत इंपॉर्टेंट बात है कि देखिए, प्लेसिबो के साथ में एथिकल इश्यूज बहुत सारे आते हैं. जब भी हम प्लेसिबो इस्तेमाल करते हैं, हमें ये मालूम है कि प्लेसिबो ट्रीटमेंट नहीं है, सबको मालूम है. आप जब रिसर्च करते हैं तो आपको मालूम है कि प्लेसिबो है, वो ट्रीटमेंट नहीं है. तो जब ट्रीटमेंट नहीं है तो हमारे सामने ये एथिकल इश्यूज आते हैं कि आप पेशेंट को ऐसी चीज क्यों दे रहे हैं जो ट्रीटमेंट नहीं है. तो आप ऐसा ट्रीटमेंट ऐसा किसी भी उस कंडीशन में नहीं कर सकते हैं जहां पे स्टैंडर्ड ट्रीटमेंट अवेलेबल है, या जहां पे कोई लाइफ थ्रेटनिंग कंडीशन है, या जहां पे इमीडिएट इंटरवेंशन की जरूरत है. वहां प्लेसिबो कतई नहीं इस्तेमाल कर सकते, क्योंकि वो पूरी तरीके से अनइथिकल है, वहां पर ट्रीटमेंट चाहिए. तो आपको क्या करना पड़ेगा, आप ऐसे में क्या करिए, उसको एक स्टैंडर्ड थेरेपी से कंपेयर कर लीजिए, या किसी दूसरे एक्टिव कंट्रोल से कंपेयर कर लीजिए. आपको कंपैरेटिव एफिकेसी मालूम हो जाएगी, आपको ये पता लग जाएगा कि आपका ट्रीटमेंट बेहतर है कि उनका ट्रीटमेंट बेहतर है. तो ये ज्यादा बेहतर तरीका होगा, ज्यादा अच्छा तरीका होगा। आपको कुछ नहीं करना, कोई प्लेसेबो नहीं बनाना, ठीक है।
Sir, that question I am trying to again put here. Ishwar Sharma has trying to ask about the Rog Nidan principle, like Nadi Pariksha. In which category we can put it in clinical practices?
नहीं डॉक्टर साहब, अभी तो इस पे डिपेंड करता है ना कि नाड़ी परीक्षा में क्या देख रहे हैं आप, क्या जानना क्या चाहते हैं, आपका आउटकम पैरामीटर क्या है, आप देखेंगे क्या नाड़ी परीक्षा में। जो आपका रिसर्च डिजाइन तय होगा। नाड़ी परीक्षा में आप क्या देख रहे हैं, आप कुछ क्वांटिफाई कर रहे हैं, उसको आप मेजरमेंट कर रहे हैं किसी चीज का, आप देख क्या रहे हैं, आपके जो आपने जो ऑब्जर्वेशन के पैरामीटर्स बनाए हैं उसके आधार पर तय होगा कि आपकी
और डायग्नोस्टिक पर्पस।
नहीं, डायग्नोस्टिक तो है, लेकिन आप नाड़ी परीक्षा में क्या देखेंगे? आप वात पित्त कफ देख रहे हैं, कि आप उसका एम्पलीट्यूड देख रहे हैं, कि आप उसका फ्रीक्वेंसी देख रहे हैं, कि आप देख क्या रहे हैं? अल्टीमेटली आप क्या ऑब्जर्व कर रहे हैं? नाड़ी परीक्षा माने क्या? जब तक आप ये तय नहीं करते कि आप स्पेसिफिकली क्या वॉच कर रहे हैं, आप कैसे रिसर्च डिजाइन कैसे तय करेंगे आप उसमें? यही तो मैं शुरू में बात कर रहा था आपसे कि आपको एक बहुत प्रिसाइज क्वेश्चन चाहिए, दिमाग में बहुत साफ सुथरे ढंग से बनाइए कि आप ढूंढना क्या चाह रहे हैं, आप कर क्या रहे हैं। तो ब्लाइंडली ऐसे ढूंढेंगे तो रिसर्च डिजाइन नहीं बना पाएंगे आप ब्लाइंडली जब तक आप ऑब्जेक्टिव्स अपने डिफाइन नहीं करेंगे। जैसे आप अगर पीको एनालिसिस करते हैं तो आपको बहुत साफ सुथरे ढंग से ये पता होना चाहिए कि आउटकम पैरामीटर हमारे क्या है, या हम इवेल्युएट किन किन पैरामीटर्स पे करेंगे। तो वो इवेल्युएशन के जो पैरामीटर्स होंगे वही बताएंगे आपको कि भाई ये कैसे मिलेगा, आपको कौन सा डिजाइन लगाओगे यहाँ पर। अगर आपको दो ट्रीटमेंट के बीच में कंपैरिजन करना है तो दूसरा रिसर्च है, अगर सिंगल ट्रीटमेंट को देखना है, आपको प्लेसेबो से कंपेयर करना है, दूसरा डिजाइन है। तो डिपेंड करता है कि आपका साधन क्या है, आप करना क्या चाहते हैं, आपके ऑब्जेक्टिव्स क्या है, उसके आधार पर तय करिए कि कौन सा डिजाइन आपका लगेगा यहाँ पर, है ना।
उन्होंने और स्पेसिफाई किया क्वेश्चन को कि दोष वृद्धि और क्षय वो देखना चाहते हैं। दोषों की वृद्धि और क्षय
देखना चाहते हैं तो दोष वृद्धि और क्षय को क्वांटिफाई करने का कुछ तरीका ढूंढना पड़ेगा। मतलब ये तो बहुत सीरियस काम है, वो आसान काम नहीं है। उसको आपको सीरियसली देखना पड़ेगा कि अगर आप क्वांटिफाई करते हो तो क्या तरीका है, आप क्वांटिफाई कैसे करोगे, क्योंकि सब्जेक्टिव पैरामीटर है वो। आपने नाड़ी पे हाथ लगाया, आपने कहा दोष बढ़ा है, तो कैसे पता लगेगा दोष बढ़ा है? आपने कहा दोष घटा है, कैसे पता लगेगा दोष घटा है? और हम क्यों माने आपकी बात, आपको हम क्यों कहें कि दोष घटा है? भाई हम कहें नहीं बढ़ा है, तो आप कैसे तय करोगे कि नहीं दोष बढ़ा है कि घटा है? तो ये तो जब तक आप एक ऑब्जेक्टिव पैरामीटर नहीं लाओगे उसके लिए ये तय ही नहीं कर पाओगे। तो आपको साफ सुथरा कोई क्राइटेरिया ढूंढना पड़ेगा। और या तो आप क्या करिए, आप ऐसी रिसर्च अगर करना भी चाहते हैं तो हाँ बहुत बढ़िया है, आप उसको मेजर करने का कोई एक स्टैंडर्ड तरीका बना लीजिए। आप बता दीजिए भाई कि इतना है, हम देखो ऐसे फील करते हैं, नाड़ी पे हाथ रखते हैं, ये पता लगता है तो हम इसको वात मानते हैं, ये पता लगता है तो हम इसको पित्त समझते हैं, ये पता लगता है तो कफ समझते हैं। और आपने क्या किया, आपने इस तरीके को अपने 10 साथियों को बता दिया, 10 लोगों को, हमारे साथ में 10 लोग हैं, हमने 10 लोगों को बता दिया। और हमने एक एक्सपेरिमेंट प्लान किया कि भाई इस तरीके से हम नाड़ी का परीक्षा करेंगे, आप भी करना, आप भी करना, आप भी करना। हमने सबको देख लिया कि भाई मरीज वही था, एक मरीज था, हमारा एक सैंपल था, हमने नाड़ी की परीक्षा करी, हमने आपसे कहा आप भी देख लो, आपसे कहा आप भी देख लो, आपसे कहा आप भी देख लो, सबको तरीका हमने वही बता दिया। तब हम देखते हैं जरा आपने क्या देखा, आपने क्या देखा, आपने क्या देखा। अगर वो सबके ऑब्जर्वेशंस एक जैसे आते हैं, हमको जो दिखाई दे रहा है वो आपको भी दिखाई दे रहा है, वो इनको भी दिखाई दे रहा है, इनको भी दिखाई दे रहा है, तो इसका मतलब कि आपका तरीका ठीक है, सारे लोगों को एक जैसी चीज दिखाई दे रही है। तो वो एक स्टैंडर्ड पैरामीटर हो गया, आपका स्टैंडर्ड ऑब्जर्वेशन का पैरामीटर हो गया, हम उसको रिसर्च में अडॉप्ट कर लेंगे। तो पता है ज्यादातर लोग उस चीज से सहमत हो जाते हैं। और अगर कहीं आपने क्या किया कि हमने देखा वाह, तो दूसरे ने कहा नहीं पित्त, तीसरे ने कहा कफ, तो फिर बेकार हो गया, फिर आप कर ही नहीं पाओगे उसको, है ना। तो फिर आपको ये स्टैंडर्डाइज करना पड़ेगा। तो ये सबसे आसान तरीका है, आप यहाँ से शुरू कर सकते हो इसको।